Okay, so good afternoon to everyone. Welcome at the next zoology seminar. And today I was asked by Honza to introduce our today's guest, who is Niloofar Alaei from State Museum of Natural History in Stuttgart. And Niloofar is originally Iranian, so she did a PhD at University of Mashhad in northeast Iran, and then she continued as postdoc at University of Vienna for a couple of years, and now she has a position as bioinformatician or evolutionary geneticist at museum in Stuttgart. And Niloofar is working on speciation genomics, if I can say, or evolutionary biology in general. And her main models are birds, but not only birds. And she's now currently working also on slugs and other organisms and in relation to their invasions. But I think today's talk will be mostly on birds.
Yeah.
So the floor is yours.
Thank you. Hi, everyone. Thanks for the nice introduction. Yeah, I'm so happy to be invited by you here and also to share some part of my favorite part of the research to you, which is about the birds, and also some very fresh and hot results that I'm so excited and I'm really happy to share with you. First of all, I want to just dedicate this talk to my friends in Iran and to the researcher in Iran that with support and collaboration make all this possible for me. Yeah, I couldn't talk more, sorry. Yeah, let's back to the science. When we see around of us and then look at the biodiversity, as evolutionary biologists, always this question comes to our mind that how these species evolve, and which process just formed them, and how they can persist through the evolutionary time. And if we want to think about the speciation as a continuous process, we can see that there is a continuous process that make one population evolve at the two separate populations, that they reproductively isolated. Speciation as a continuous process that during this process, two populations can separate, isolated for some time, merging again and maybe separated. At the end, maybe we can have some reproductive isolated population that we can call them good species. Yeah? When they come into the contact, they even cannot mate. But the thing is that there are different kind of differences and incompatibility that accumulate during this speciation process, that at the end we can have the two species. And theoretical and empirical data shows that behavioral differences can maybe evolve at early stage of the speciation, and sometimes they also claim that can initiate the speciation process, such as the behavioral differences, such as the mating signal and female preference, yeah, that lead to associative mating. To know more about this speciation process, that how a species form and how they persist, we can use this time of the speciation, yeah, the times that when two young populations come into the contact. This merging area that called hybrid zone give us a very great opportunity as evolutionary biologists to just narrow down to see that what's happening during the speciation process, how these two species can be distinct. And if we want to take a look, quickly look at what's happening in the hybrid zone, in this example, you can see that, for example, I have a bluebird and redbird, they come into the contact and they generate the hybrid. The thing is that most part of the genome can be homogenized by the gene flow, yeah? We can see that in the hybrid, most part of the genome is just homogenized, except the part that the gene flow cannot happen there. We call this area as a barrier genes, that here in this example, these barrier genes associated to color pattern. And this gives us the power to find these barrier genes. Why? Because most part of the genome show it just homogenized, show very low genetic differentiation, but except the part that the gene flow cannot affect there. Yeah, and if we want to compare with the situation that we have two species that cannot hybridize, we can see that what's happening. In this case, We have, again, two birds with a two different color pattern, but almost most part of the genome shows the high genetic differentiation, because we know that different process can make a differentiation. It could be just by random. It could be it doesn't relate it to any significant for the speciation, yeah? Then it's really important, this hybridization gives us a very great power to find this area. And it's the reason that they call hybridization as a natural laboratory. As here, we can see that in the hybridization, we can distinguish these two parts of the genome from the neutral genes. That we know that in the last example, the neutral genes is most part of the genome are neutral, and they just homogenize. The gene flow can freely happen there. And then we have a power to find the barrier to gene flow that the gene flow cannot happen, or some asymmetric gene flow region that mostly gene flow happen in one direction, that we expect some adaptive genes is just introduced from one species to another species. One approach that we can use to identify this part of the genome is the Klein analysis. In the Klein, in the y-axis, you can see that the frequency of species B is not a matter that which character you use. For example, you can say the color pattern, yeah? Is the red bird and blue bird. You can say is the genetic traits. And in the y-axis is the geographic distance. Here you can see that in the hybrid index, we have a red bird, and we have a blue bird, yeah? The alleles that is related to the red color and the blue color. If this area is the barrier genes, we have that. It's just red allele, and then it's a very sharp transition to the blue allele. And then the width is really narrow. It shows that in that area, that is the geographic distance, for example, the hybrid zone, the center of the hybrid zone is in the center of the Klein. Yeah? In that area, we don't have a lot of individual with the intermediate hybrid index. It's only one parent or another parent. Why? Because gene flow cannot happen there. But if you compare it with the neutral genes, you can see that the width of the Klein is just increased. Why? Because the gene flow can happen, and you have the hybrid index of one parent, some intermediate hybrid index, and then hybrid index for another parents. And then the change of these two alleles, you can see that is really shallow compared to the barrier genes, which is really steep. And we also have another scenario that is the asymmetric gene flow. It's again, you can see that it's the very sharp transition because this area is under the selection, but the center of the hybrid zone is just changing because it's mostly the gene flow happen from one parent to another parent. Yeah, it's not a symmetric gene flow. And if you also want to think about any kind of genetic differentiation, for example, FSD, we can also expect that in total, the genome average should be really low because most parts of the genome are neutral and they just homogenize, except the parts that we don't have a gene flow there. Yeah? Then we expect to see such an increase of a differentiation in a region that cannot homogenize by the gene flow. That one of the most common things or a statistic that they use is the FSD, which is the genomic differentiation between the two populations. And now let's back to the color in nature. Color is one of the most, at least for me, dramatic change that you can see when you look at the sky and you look at any kind of animal, you can see the different colors. And it's really functionally important trait that can be affected the speciation in a different way, yeah? Through the camouflage, through the mimicry, mating behavior, and even the foraging behavior. Okay, yeah. The foraging behavior, and also not only in the animals that now we have around of us. Recently, our colleague in the Natural History Museum of Stuttgart published a paper in the Nature that they found a new fossil, that they even found the melanogenes in the fossil that shows that the color also exists in that fossil. Then we know that even in the animal that is already extinct, we have a coloration. We don't know still that what's the function of the coloration, but we have color, even in the extinct animals. Yeah, then we can use this hybrid zone to found the genomic architecture of this functional trait, and also to found the genomic sources, that what is the sources of this huge biodiversity that we can see in the color pattern around of us. Here at first, I want to share the result of a very long-term collaboration with the group of the people in Switzerland, that we worked in a group of the birds with the black and white coloration, and last year we can publish the result in the Science journal. And here you can see that is a phylogeny based on the whole genome data for the wheatears and some other related genus, that they shows the extensive convergent evolution. At the top, you can see that is the migration behavior, sexual dimorphism, and the throat color. And this trait is always changed across the phylogeny. Yeah? And for this research, we mostly focus on this group of birds that called Hispanica complex, that they show very convergent evolution in the very strong convergent evolution in the throat color. This group includes 5 different taxa that, based on the whole genome data, we can reconstruct this species tree topology. And out of these 5, 3 of them shows the color polymorphism. Yeah? They have a polymorphic in the one population, in the throat color and in the mantle color. Yeah, we have a black throated and a white throated. At least 3 of them shows the polymorphic. And they also show the extensive convergent evolution, means that this guy, based on the phenotype, is really similar to the Peleshanka and Pontic, but is the sister taxa for Melanaloeca. At the same here, Hispanica, based on the morphology, is really similar to Melanaloeca, and for a long time they considered them as a 2 subspecies, but they even not a sister taxa. Yeah, then we can see that here in this group, the color pattern cannot reflect at all the true evolutionary history. And this black color is just evolved independently, maybe, we didn't know yet, is independently or dependently through the phylogeny. The cool thing is for this system is that we have several hybrid zones, that in the north part of Iran and around of the Caspian Sea, and then western side of the Black Sea, we have the hybrid zone. And this hybrid zone, this bird shows the different color pattern, all the intermediate color pattern that is possible. Through the black throated, black mantle, neck, it's a really combination of the different color pattern. Then we expect that there should be a lots of gene flow happen in this merging time, and it helps us to narrow down the part that can associate to this convergent evolution or to this color pattern. Our data shows, our analyzers, we use the whole genome data with the, this, you can also see all the samples that we use. We try to cover most part of the distribution. And our data, our analyzers shows that one region in chromosome 20 can explain this color pattern in the throat. Yeah? And when we narrow down, we can see that it's the one mutation in a protein coding genes that's called ACIP, and also one insertion, a region with 9,000 base pair is just inserted in here, that we don't have it in a black throated. Means that it's a combination of some regulatory change and also some protein coding change. And if we look at the 2 birds with the black-throated and the white-throated in a different part of the body, and look at the expression of this gene, we can see that in the white morph, everywhere that we have a white color, the expression of this ACIP gene is just increased compared to the black color. That this ACIP genes just has the inhibitor effect on the MC1R that generate the melanin, and melanin is the pigmentation for the black color. Yeah? And if we look at the evolutionary history of this color polymorphism in the throat, we can see that our red data shows that the black-throated is an ancestral morph, and the white morph, this mutation happened around 0.5 million years ago, and for the first time introduced the white-throated in a Melanoleuca. And then this mutation is just introduced by integration to the Hispanica and Peleshanka. Yeah? Then the genomic basis of this convergent evolution that at the throat color is just a mutation that happened in one species and then introduced through the integration to other species. And now the question is that it's really the adaptive integration, means that can we think about that this mutation brings some adaptation, some advantage for the Melanoleuca for the first time, and it's the reason that it's just introduced to other species. It's the bird and it's the color. Then the first thing that comes to our mind is the sexual selection. We are thinking that maybe it's some differences, or for the female is more important that the throat is white or black. But it's not the case. In the field, we found that for the female, it's not a matter that the male is a black-throated or white-throated. Then we studied the nitrogen stable isotope in the blood, and we found that black-throated and white-throated using the different kind of insect. Means that this mutation associated with the foraging behavior. They are living in a very open and arid habitat, and then using the different kind of food help them to decrease the competition. Means that this mutation that brings the white-throated morph, indeed, under the natural selection, not sexual selection, because help them in the harsh type of the habitat that they are living, there is less competition for the food. Yeah? They can use different kind of food resources. Then we can also, I think it's there to say that it's a really cool example of adaptive integrase genes. Means that this mutation brings some advantage and then introduced to other species through integration. But it's not always the case. Here in this example that we saw, we see that the genes that is associated with the color patterns show some pattern of the convergent evolution. But it's not always the case. I want to show another example of our very recent research that we got the DFG, we got the DFG grant for this research last year. And now Louis, our PhD student, started since 1 year ago, started working on this project. It's exactly another side of the coin. Means that we found the genes, maybe, which is associated with the color pattern, and these genes show the divergent selection, divergent evolution, not convergent evolution. These genes may be associated with the coloration, just distinct these two species from each other. Again, I want to just introduce this system to you. It's the black-headed bunting, red-headed bunting. These two birds are phenotypically totally different. The males show different color pattern, and they meet in northeast of Iran. They generate the hybrid with the intermediate phenotype and also some transgressive or novel phenotype that we can only see it in a hybrid zone. Yeah? Our colleague in 2023 studied the based on only morphology. They show that the center of the hybrid zone since 50 years ago moved toward the west. And they make this hypothesis that maybe recently by the global warming and land use change, more suitable habitat provided for red-headed bunting. Red-headed bunting expanded distribution and pushed everything toward the west. Yeah, but it's just a hypothesis based on the morphology data. Then for running this project, at first we are asking this question that if these two species are ecologically diverged or not. Our colleague Sajjad did the niche modeling analysis using the GBIF data for these two species. And at first we just looking at we use the current bioclimatic layers that is available. And if we look at the data, we can see that it's higher suitability provided for black-headed one compared to the red-headed one. And the ecological preference of these two species are significantly different. The black-headed one is just prefer mostly wetter and the warmer environment, although the red-headed one prefer drier and colder environment. And we repeat the same analysis by using the bioclimatic layer for last glacial maximum. And our result, when we compare it to the now, we can see that most of the species, both species just likely experience a range expansion. And the range expansion after the glacial means that now here in the current interglacial period is a bit higher for the red-headed bunting. Red-headed bunting experience more expansion compared to the black-headed bunting. And then the next step, we want to look at how this species form. We use the whole genome analysis, whole genome data, and then I use the fresh sample and the museum sample to cover most part of the distribution, to have both from the allopatry and from the sympatry. I include 7 samples from the intermediate phenotype and 1 novel or transgressive phenotype from here. The first thing that we just reconstruct the mitochondrial tree based on the whole mitogenome. And the divergence time based on the mitochondrial data is around 2 million years ago. And if we look at the whole genome data or nuclear data, and that I reconstruct the demographic analysis, that if I want to go through this, is we have ancestral population, and around 0.5 million years ago, the speciation happened, and the effective population size of the black-headed bunting is much higher than the red-headed bunting. And they come into the secondary contact, and it's a very strong asymmetric gene flow from the black-headed to the red-headed. The strange thing is that if you look at the time of the divergence between these two species based on the nuclear data is 0.5 million years ago, and here is 2 million years ago. It's a very big differences. Yeah? But the cool thing is that our demography result is really well matched with this hypothesis that our colleague made in 2023. Yeah? Our data shows that they have 2 different ecological preferences. And our result also shows that there is a very strong asymmetric gene flow from black-headed to red-headed. And after secondary contact, we have more expansion in a red-headed one. Then it's more likely that recently we have more suitable habitat provided for the red-headed one. And red-headed one expand the distribution. And if we want to believe this scenario, then we expect this such a huge amount of the gene flow from the native species, let's call it the invasive species. Yeah? Then, based on our data, then we will have a huge amount of the gene flow. We want to see that what's happening after this secondary contact. What's the admixture pattern in the hybrid zone? Then here it's the admixture pattern. That red color shows the ancestry of the red-headed one, and the blue color shows the ancestry of the black-headed one. And you can see that in the allopatry, they are almost pure. And in the hybrid zone, we have a different kind of admixture pattern. Yeah, we have some sample from middle of the hybrid zone that looks F1 or F2, and also some sample with a different proportion. And the cool thing is a transgressive or novel phenotype that shows some back crosses with the black-headed one. Because when I always think about the novel phenotype, it comes to my mind that, okay, it could be from the first generation, and it's just the 100% heterozygosity, then we expect some novelty comes up. But it's not the case, yeah? It's just some generation back crosses from the black-headed one. And as I show you, what we expect when we look at the genomic differentiation. Yeah? We expect that the genome average should be low because most part of the genome are neutral and they homogenize. And we have some clear peak that shows the high amount of the genomic differentiation. And our result true result is also shows more dramatic situation. You can see that the genome average is almost close to 0, 0.06. And we have a very clear peak of the high genetic differences. For example, here in the x-axis, you can see the different position in a chromosome 2, and in the y-axis is the FST. Most part of the chromosome 2 shows very low FST, except the 2 parts that show very high peak of the FST. And this gives us the power to find this part that could also the barrier against the gene flow. And it's not the pattern only for chromosome 2. If we look at the other chromosome or a scaffold across the genome, we can see that it's the same. For some of the scaffold or chromosome, we have some very clear peak, but for most of them, most part of the scaffold show very low FST. And I can say that is only 1.5% of genome shows genomic differentiation above the genome average. Maybe this only 1.5% of the genome, we can just make it link with the barrier against the gene flow. And now, if we back to the part that why we see this such a big differences in the time of the divergence between mitogenome and nuclear genes, we can think that what we expect to see in the barrier to gene flow regarding the coalescence time. Most part of the genome is just homogenized by the gene flow, except the part that the gene flow cannot happen then. There we expect to see the deeper coalescence for that part of the genome compared to the rest part of the genome. For the mitogenome, we estimate the time of the divergence 2 million years ago. We have a 2 well-separated clade that is just separate the whole red-headed from the black-headed one. Compared to the neutral part of the genome or the nuclear genes, that they have a very shallow coalescence time. It's around 0.5 million years ago. And I just extracted some part of the genome with the high genomic differentiation. And the coolest thing is that they show a very similar pattern to the mitochondrial genes. They show the deep divergence as we expected for the barrier to gene flow. Then when I look at this result, the first thing that comes to my mind is that maybe mitogenome is under the selection also, because the mitogenome shows the similar pattern of the barrier to gene flow. And this also explains that why when we look at the whole genome, we can see the very young speciation time, because as I showed you in the last slide, more than 90% of the genome shows very low FST. Means that more than 90% of the genome is just homogenized by the gene flow. Then I think here we have a system that only limited number of the genes just separate these two species. We have some genes that is important for these two species, separate them, make them distinct, and the rest of the genome is just homogenized. To answer that, what are the barrier genes? Here we saw that we have, Here we saw that we have some barrier genes, especially in this. We have some area with a very high FST. Then the question is that what is the barrier genes? To answer this question, we need more sample from the hybrid zone. And then last year, I went to Iran, and with my colleague, my friends there, we did the sampling. We got the bird with a different color combination, and you can see that this guy is from the Greece, is the pure one, and you can see that how this color pattern can be different in the hybrid zone. It's amazing because each bird that we catch shows the different color pattern. And the color pattern can be mixed in a very, very different way. It's one of the kind of suitable habitat for that bird in the northeast of Iran that we can even see the chicks there, is there. And if you compare it to Greece, because at first we went to Greece to do sampling for the pure part of the distribution, you can see that how differences in the type of the habitat between these two species. Yeah? And also, the differences is around 3-4 days. Here we still have the eggs, but here we have chicks. Yeah? It's totally different breeding season. They started much earlier in Iran because it's much hotter. Because it's hotter, yeah. And then for catching this bird, I just put some picture of the field work to don't make you so sleepy during the talk. It's the mist net that we play the song of the bird, and because it's the breeding season, the male becomes so aggressive, want to defend the territory and his own female, then they just come into the mist net because they become just simply crazy. Otherwise, if it's outside the breeding season, they can see the nets. And, oops. Yeah. When we catch the bird, we just take a photo in some standard light box to be sure that at the end of the day, we can compare all these color pattern in a different side of the birds. We get the blood sample, we measure the different morphometric traits. And then at the end, we can increase the number of the samples to around 130. And we have some sample from Afghanistan, from Kazakhstan, which is from pure site of the distribution of red-headed bunting. We have sample from the Greece, Turkey, Italy, and Russia, which is the pure sample of the black-headed bunting, and we have more than 90 sample from Iran. And you can see that here is the center of the hybrid zone in the north part of Iran. And the more crazy things is that even in the middle of the hybrid zone, you can see the pure sample. Yeah. Again, this color, the red color, shows the ancestry of the red-headed bunting. The black color shows the ancestry of the black-headed bunting. And in the middle of the hybrid zone, we have a hybrid with a different ancestry proportion, if you look at the pie chart. But even with the hybrid, we have some pure parents of red and some pure parents of the black. To me, it looked like that the hybrid is under the selection. Because if I want to compare it with the last case study, which is the wheatear, for the wheatear, whole Iran is just a hybrid. They just hybridize crazily. But here we can see that the hybridization is somehow limited, even in the middle of the hybrid zone. And to just check, have a first checking that how strong is a reproductive isolation, we use this kind of analysis that compare the hybrid index to the heterozygosity, that's called triangle plot. In this x-axis, I have a hybrid index that, for example, for the bluebird, the hybrid index could be 0, and the redhead shows the hybrid index of 1. And the F1 hybrid shows the hybrid index exactly in middle because got half of the genome from one parent and half from another parent. Then the hybrid index should be 0.5, but the heterozygosity is 100%. Yeah? Then there are several possibilities. The hybrid can just mating together and generate the F1, Fn. In that case, the hybrid index should be, again, around of 0.5, but the heterozygosity decrease because more recombination happen. But the hybrid could also have back crosses, yeah, with the parents. In this case, the hybrid index goes toward the parents that the hybrid has the back crosses. Both heterozygosity and hybrid indexes just goes toward that parents. Yeah? It could be has some back crosses with the black-headed one, then the hybrid index goes toward the 0 and heterozygosity decrease, or could have some back crosses to red-headed one, then in this case the hybrid index goes toward 1 and the heterozygosity decrease. Yeah? And what we expect, if we have a very free gene flow in the hybrid zone with no selection, we expect most of the hybrids should be in between, in the middle. that shows that, for example, it's the case for the wheatear project, because they shows that they can mating together, they can mate with the two parents. Then in that case, we call it hybrid swarm, because they can freely mate together with the parents and then come together. But look at our result. The black color is all the sample from Iran. Red one is the red-headed bunting from the outside Iran, and the black is and blue is the black-headed bunting from outside Iran. And you can see that we have very little number of the sample in the middle. Most of the sample shows back crosses. Yeah, we have maybe 2-3 sample of F1, F2, and then the samples wants to mate with their own parents. And the cool thing is that I just color them based on the 2 mitochondrial clade that I have. Yeah? And we can see a very clear separation. Means that red dot get the mitochondria from the red-headed bunting and nuclear also from the red-headed bunting. Means that the samples that get the mitochondria from the red prefer to also mating with the red. And vice versa here. This guy, for example, here, based on the mitogenome, get the mitochondria from the black-headed one. And based on the nuclear, also shows back crosses with the black-headed one. To me, it could be the signal of the very strong assortative mating or mitonuclear incompatibility. Means that the F1 hybrid that get the mitochondria from one parent, it's something against and don't like to continue mating with hybrid or with another parents, want to have their own father from the same parents. Yeah, it could be also some signal of the mitonuclear discordance, but we should work more on it. And then, yeah. Here, as I told you, also there are also another way that we can see that how much selection play a role in the hybrid zone. Here we define some morphology trait, for example, the head color, the collar, the back, the rump, and also 3 morphometric traits that can separate black-headed one from the red-headed one. And we look at the Klein analyzers for each of these traits to see that if the Klein is really steep, means that it's under the selection, or no, the Klein is really shallow. That means the gene flow happened. And it's the result that also looks so promising. All these very steep Klein are based on the morphology, means that all the traits that we define based on the morphology, for example, the head color, everything looks very strong selection, or looks the signature that they are under very strong selection. And these 3 traits are the morphometric trait, which is show very shallow, which shows that the gene flow that just homogenized them. Yeah. But the cool thing is that it looks that all the evidence together that the sexual selection can play a very important role in this hybrid zone. Yeah? And somehow the hybrid with the intermediate phenotype, maybe they are not so sexy for the others. They don't want to, birds don't like them to mate with them, or they show some incompatibility at the genomic level. And here is the Klein analysis based on the whole genome data. You can see that we have a very steep Klein for the purple one, and we have also some Klein, some genes that the center are the same, and they show some asymmetric gene flow, yeah, in both sides. Then for sure, we have some part of the genome that shows very clear signal that they are under the selection. And the Klein is really steep for that area. This part is a very, very hot and new result that I want to share with you. As I told you, with my PhD student Luis, we went to Greece last year to do the sampling for our own reference genome. Because all the analyses that I show you, I use the reference genome that already published in the NCBI, is not closely related to my species, and the quality also is not really high. Then we make our reference genome, and last month we received the reference genome. And I repeat or Lewis repeat all these kind of FST plot analysis, yeah, by using our own reference genome. And we hope that we should get a better result because it's really closely related to ours. You can see that here I just put one of the chromosomes, is chromosome 1. Again, the genome-wide FST is really low. Here is the genome position of the chromosome 1, and y-axis shows the FST. The red line shows the genome-wide FST, which is really low, and we have some region with a very high peak of the FST. Yeah, it's the things that we already see in the last result. But the cool part is the chromosome Z, which is the sex chromosome, and we can see the pattern that shows, at least to me, is the inversion pattern. It looks that some inversion happened in this group of birds in the Z chromosome that most likely included the sexual genes. Yeah? And this, again, put another layer of the evidence that insists that in this system, the sexual trait is really under the sexual selection. Or could be sexual or natural selection. I can say that the sexual traits are under the selection. We still don't know which type of the selection play a role. But this is super promising to me because it just looks like the inversion that happened in the chromosome Z. And if you compare the pattern of the FST in the chromosome Z, you can see that most of the chromosome Z shows the higher FST compared to the autosome. And if you, in the lower plot, you can see the pi, the genetic diversity within the population. And here you can see that the genetic diversity of the black-headed one is just increased in that part of the genome. Could also reflect that maybe the black-headed one has some inversion and the red-headed one has the normal. And if it's the case, we need to do more analysis, but if it's the case, it just also explains that why this hybrid zone is under a very strong selection. We cannot see a lot of F2 and F3 hybrids. Yeah, but it's under the progress, this part. I want to shortly talk about the future parts, future paths of this project. One of the main aims of this project is to find the genes which is associated to color patterns, and we expect these genes should be under the very strong selection. And then it's some preliminary data that I run DEEM for them for the chromosome painting or polarization. And here I put all the hybrid together as a one group as a hybrid. But you see that the hybrids show differences in a different trait. Then in the next part of the project, at least we use our own reference genome, and we want to find that if this area with the high peak of the FST could also reflect the color pattern or not. Can we just say that all the sample with the black-headed, for example, are homozygote for one parent, and all the sample with the red head are homozygote for another parent, and the hybrid with the intermix show the heterozygote position or not? Yeah? It's one of the aims of this project to see that if the region that associated with the color patterns is under the strong selection or not. And also, when I think about this system and compare it with the wheatear, which is only black and white color, it always comes to my mind that maybe what we see and we simply link it to the sexual selection is not only sexual selection, because this bird has the both melanin and carotenoid. And we know that carotenoid is mostly coming from the food. Then it's also not more likely, but it's possible that some part of the color pattern that we see in this bird is just because of the ecological selection, not sexual selection. Then at first I did the spectrophotometry to be sure that we have a carotenoid-based color that we have. And now I'm thinking about this. If really only behavioral differences or sexual selection just play a role, or both ecological and behavioral, it means that we have two young species, lots of gene flow happen, but they are really distinct compared to the wheatear project. Yeah? They are really distinct, and the selection is against the hybrid in somehow. then to me, sexual selection maybe alone cannot just provide such a strong pattern. Maybe the combination of the sexual and natural selection. And then I think we have enough evidence. For example, we have some trait which is under ecological selection. Our ecological modeling data shows that these two species are significantly different from the ecological preference. The red-headed bunting that comes from the Central Asia prefer hotter environment compared to black-headed one that comes from Europe. We have some trait that associated with the color patterns that show the significant differences between these two species in allopatry. And we have some trait, for example, such as the bill length, that could associated to the food also shows the significant differences between black-headed and red-headed. Then I think we have enough evidence to say that maybe ecological selection also play an important role in this group. Sorry. Then fortunately, we also, when I did the sampling, we sampled also the poop of the birds. And then now I'm thinking for the new project, we can use this poop, do the environmental DNA to look at the food composition of these two birds, both in allopatry and also sympatry. Because the cool things for this system is that we can even have the two pure parents in the sympatry. Yeah? Then we can compare the food in the allopatry and in sympatry, and we can see that if we find any significant signal of the different food, especially in a sympatry or not. Because if we can find it, we can map it to the reference genome, and we can find the part of the genome that may be associated with this ecological preference of the food. Yeah? And I can imagine that there is two scenarios that can play a role here. One could be magic trait, means that, for example, just imagine, we find the part of the genome that associated with the color genes. We found the part of the genome that can associated with the ecological adaptation. And when we compared these two, we found that both are in the same gene. Then it could be the magic trait, the trait that associated with the sexual trait and the ecological trait. Or in another scenario that I like it more is that we found some different genes, and these different genes is just totally linked together. It could be physical linkage or it could be a functional linkage. Yeah? Means that when we have the red-headed bunting with the red color, we always have this kind of food preference. They can maybe associated with the different genes, but always linked together, because red just have this food. It's the idea that now we are working to write a proposal for the Humboldt that my friend from Iran can join our group in Stuttgart, and we want to focus on here. That if only parazygotic barrier, which is the sexual selection, play a role, or no, we can have both postzygotic and parazygotic barrier, because we, in the species continuum, we are here. We have a very two young species that can hybridize together. Yeah? Means that the speciation is still running. And then we can focus on which kind of the barrier is just accumulate in these two group of the birds. And the last part that I want to show you is that we also have the museum sample for these two group of birds. They belong to 50 years ago, and they are in the Museum of Konig in Bonn in Germany. That hopefully we get access to this museum sample, and then we can compare the old or 50 years ago with now. We have group of the birds that show some trait under the sexual selection, show some trait that are under the most likely the natural or ecological selection. We know that this hybrid zone is a moving hybrid zone. Then we can compare that what's happening through this changing distribution or adapting to the new habitat that human provide for them, what's happening for different kind of the barrier. If all barrier behave the same, if all of them shows moving in the center of the hybrid zone or not, is the parts that we are still working on. And at the end, I want to thank you all my collaborators in Iran and also in Europe. Thank you all the funding agency that give me this power or the money to do this research, and all of you to be here today. And I'm here for the discussion.
Thank you, Niloofar, for excellent talk, and we have time for discussion. I have a couple of questions. Stuart?
Thank you, a lovely talk. I'm wondering, and perhaps you mentioned it and I missed it, with the movement of the hybrid zone, it's gone maybe 50 kilometers, it looked like. Behind the hybrid zone, do you have any evidence that there are footprints of the genome that used to be there? Are there any genes left behind?
Yeah, I can also show you quickly. Yes, we have it, yeah? Because in the past, they believed that the hybrid zone should be just limited to that area. Yeah? And if you see, we have still some, now we don't see the hybrids here. If you go for sampling, all the samples, at least phenotypically, look like the pure one. Yeah? And the center of the hybrid zone is now here. And we have still some footprints of the maybe old hybridization that happened there. But regarding the genes, no, we still don't know that which genes is still remain there. Yeah? But at least the result shows yes. Because, for example, if I look at the phenotype of this sample, based on the phenotype, it really looks like the pure one. But based on the genotype, you can see that a small proportion of the black-headed one, that to me look like some footprints of the remaining hybridization from the past.
Thank you.
Sure. Thank you for coming. No, it's not unique. You cannot see any kind of differences, for example, between the habitat here and the habitat here. No.
So it's not, it's not ecotone there.
No, I don't think so. I don't think so. It's a transition between these two ecotone. I don't think so. It's just the region, and this area in north part of Iran is so famous for one of the hottest zone for the songbird hybridization. You can see that there's several, for example, here I show you two. I'm now working with another group of the collaborator in north part of the Germany, working on another group of the bird that has a hybrid zone in the same region. Yeah, mostly the eastern and western of the Palearctic come to each other in the north part of Iran, and then they make a hybrid zone there.
Partly answered that with this magic traits, etc. But how abundant and how linked, especially, are those peaks? I mean, of course, physically they are not direct plots, but are they linked somehow functionally? For example, the same kind of cascade for transcription and then for functional.
Yeah, I hope we can find this. It's a very young project. We still don't get to this point, but exactly because for sure some of the links, now I think I put it in my slide. Some of the links that we have for sure is just simply related to, okay, sorry, in the wrong direction. Yeah, some of the peaks that we found simply just shows the different pattern of the recombination, yeah? maybe it's just a link selection, and we found that because it just mimic exactly the same pattern of the divergent selection. And I'm pretty sure when we narrow down, we can see that some of the peak is just have nothing related to the speciation, but for some a small peak, I think, yeah, we can find some of them that maybe they're just really linked together. And I hope we can find this such a pattern by using the genome polarization in DEEM, because if we find that always homozygote for some region is under the selection linked to some specific kind of the homozygote and heterozygote in another chromosome, maybe it's just a signal of the linked, physically or functionally linked. Mm-hmm.
Here we go.
I would have a question about the former species or species complex of wheatears.
Wheatears, yeah.
You showed that even within a population you can have such with a light throat and a dark black throat.
Mm-hmm.
Do we know anything about ecological selection, about any advantage of one compared to the other? And a more broader question, you have these species which have a lot of black, then you have these kind of more sandy, light colorations. Now in desert, semi-desert habitats, very often we find species of all different kinds which are kind of have this light coloration, which might be so that they don't heat up too much, but also it's masking, it's kind of camouflage. What is the advantage of this black coloration in this hot habitat? I mean, it's kind of like a job, you know.
Maybe it's the reason. Yeah, to us also it's so surprising because these birds coming from very arid adapted, it's just totally arid adapted birds and coming from very arid and open areas. And when we went for the sampling, you can't find the female because female coloration is totally sandy, but you can find the male so easily because just imagine in a gray kind of habitat, a small black thing just start flying, yeah? Then honestly, I have no idea that why all these birds, it just shows black and white coloration. But regarding your question of the black-throated and white-throated, yeah, I think it somehow is the example of the balancing selection, means that in one population you have both black-throated and white-throated. And even in the same habitat, you can see that the black-throated and white-throated is just making the nest really close to each other. And I think it's the one way to just decrease the food competition. Because the hypothesis that we make is just somehow copy of the hypothesis of the barn owl, that these different color patterns affected on the food that they can prey. There are insectivorous. And we think that, okay, if the throat is white, maybe amount of the light that they can just reflect are totally different to the birds with the black-throated. And it affected on the different kind of insect that they can prey easily. And to me, it's just some case that these two birds live together in a harsh habitat and they use the different food resources. Sure.
So a question about those FST peaks. Just an idea. You showed that in coloration you have different phenotypes, like mosaic of different features. So it came to my mind that this phenotypic mosaic can correspond to some genotypic mosaic, some distinct combinations of genotypes in these peak regions.
Yeah, that's true. It's also one hypothesis that we think that even if we find several genes, maybe these genes is just interacting together in an additive or epistatic way. Yeah, means that, for example, if this gene is somehow expressed and this gene is not expressed, we can have the yellow with some patch of the black. And if we both express, we can have totally black. And if we both not express, we can have the red. I think it could be one hypothesis that more likely happen that there are several genes that just interact together in a different way, but we still need to discover in that part. Sure, thank you.
I'm thinking about what you showed, differences in time estimation of divergence according to the marker used. So does it mean that for proper time estimation, we should use just those barrier genes? Because the others are kind of homogenized or populations in between them. Yeah? So, for example, you showed that the mitochondrial genome looks like being a barrier to gene flow. So we can use it for proper time estimation, or just those high peak part of the genome.
Yeah, you know, to me, it depends on how you define the speciation. Yeah, because if you want to look at the one species in total, is also included the neutral part. And maybe this part, which is under the selection, is the part that they gain in the allopatry. And then when they come into the contact, if you want to estimate the genes in the allopatry, maybe it's good to just only focus on the barrier genes. But most of the time, even the nuclear is just cannot reflect the time of the allopatry speciation. Yeah, if you think about the allopatry speciation, sometimes it's sympatry and make the situation more complex. But if you think about the allopatry, for example, in the crow system that has a hybrid zone in the center of the Europe, even the mitogenome shows very shallow divergence time. And they are totally mixed. Even the mitogenome cannot separate the two clade of the species. Yeah, it really depends on the system. But I agree with you, if you want to just estimate the right speciation time, just call it right, then we can focus on the barrier genes because it just reflects the allopatry speciation. Because I can imagine that these differences is just accumulate in allopatry. They come into the contact, the gene fellow homogenize the rest part of the genome, and then the peak in the allopatry still shows the divergence time. Yeah, I can imagine that could be the scenario.
I had a question related to Martin's question. So you have not yet studied in detail these barrier genes or the genes which are on these peaks of FST.
Yeah.
But when I saw these coalescence trees, so there is the tree is similar for mitochondrial DNA and for these barrier genes. So it would be interesting to check if there are no these Oxford genes, so the genes encoded by nuclear DNA that work in mitochondria. So that makes
that would be great, because we also in the triangle plot we can see a very great in agreement between the mitogenome and nuclear genes, that everything based on the mitogenome are separated, shows the same pattern in the nuclear genes. Then in the next step we want to see that which genes are in the peak of the FST. I hope we can find some genes that are associated with the mitochondrial and also some genes that may be associated in adaptation.
Yeah.
No.
And another question completely different. So in this wheatear system, so you said it's somehow simpler because they are just black and white. But if I remember well, so birds can see even in UV part of spectrum. So they are still black and white in this. They see each other black and white.
That's true. That's true. Because I just speak based on my eyes. That's totally true. Yeah. The birds that they see, maybe for them is not a matter. I just talk about is simpler because there is no effect of the nature or the food and the color, yeah, because there is no carotenoid. We know that carotenoid coming from the nature. Then if they adapt to the different kind of habitat, they will adapt to different kind of food, and it affect on the coloration because the carotenoid coming from food. But at least in the wheatear, we don't have some color pattern that coming from food. Yeah, it's not there, but at least our result shows that it's also the throat coloration is just shows the foraging behavior.
Yeah. One more question from Stuart.
In the group as a whole, so they all look like small birds with pointy beaks to me. But I know this group as ones where, for example, in Ireland we'd spot them because they've got interesting head color. The males have interesting head color. So in that large group, is it generally considered that the females are doing sexual choice based on male head color?
Yeah.
In that large group, sorry, has there been any work to try and locate a female choice locus?
Yeah, it's a very good question because here, for example, I talk about the behavioral isolation, yeah? And then behavioral isolation is the mating signal plus female preference. But I have nothing regarding the female preference. The first thing is that because we cannot assemble the W chromosome in the birds, ZW is the female, yeah? We can, because it's so difficult, we cannot assemble it. And the second part is that most of the samples that I shows is just male because male has a different color. For the female, even when we catch it, we even cannot distinguish if it's the female of one species or another species, or if it's the hybrid. Then we only focus on a male, and I think we don't have power to find the part of the genome that may be associated with the female preference. Because in that case, for example, in a study at the cricket that for sure you know, they also can pinpoint mating signal of the male and the female preference. I think we don't have a power here because simply we don't have a W chromosome and we don't sample of the female. That's a pity.
Thank you.
Sure. Thanks.
The second system, the hybrid zone, is quite restricted in space. My question is why? And then if I got it right, it's happening in really warm climate or a specific environment. So is that the hybrid has some advantages, like higher fitness, so that's why they hybridize, because the hybrids can cope with this hard climate better. And what would happen if you took the pairs from the end of distribution? Do they hybridize? Have you ever tried?
For the part of the hybrid zone, I think why the hybrid zone is so narrow is just because at least our result shows that there is a strong selection against the hybrid. Yeah, means that it should be some incompatibility at the genomic level. It could be some assortative mating that the hybrid with the intermixed color pattern doesn't look sexy for the female don't want to mating for them. Or if they mate for them, maybe the individual is just died or is not fertile. But the thing is that this hybrid zone is under very strong selection. And regarding putting from two sides, we didn't do that, and I think we don't have a power to do that. But I guess we should see the same result. Because these two birds are the young bird, and we can see that in most part of the genome, the differentiation is really low. Means that I don't think so that we have lots of incompatibility at the genomic level. Then I guess we should see the same result. But we didn't do that. Sure.
Okay, I don't see one more question.
One is just follow up that again, if the memory serves well, the males were black, the females are like gray.
Sandy, yeah.
Sandy.
Uh-huh.
So would it be that it's related to the Zahavi's handicap thing? That if you can survive being black in such a hot weather, so then it means that your super thing is super whatever.
So yeah, superhero. Yeah, maybe. I think for the female, it's so important to have a better camouflage because they take care of the chicks. And for the male, they have nothing. They do nothing, really nothing. I was in the field, and even if I went 1 week later, means that the male is totally be sure that mating finish and the female is busy with the eggs. Even the male doesn't want to spend energy for defending the territory. Really did nothing. We saw the birds, and then the female is just feeding the chicks. And maybe in the very hot temperature, 41 degree, the female is just going back and forth to catch the insect, and the male do nothing, just sitting on top of the tree, even don't defending, even don't respond the song. Then I think the only things that the male can do is just be sexy. Then the female select them. Just in the birds, not.
It's very familiar to me. It's a very female perspective. You just don't appreciate the work just sitting up there.
Yeah.
Is it hard to find the nest with chicks?
Yes, yeah. Even it's really hard to follow the female because female is take care a lot. When she's coming out of the nest, she tries to sit in several different places to be sure that no predator is just follow her. Yeah, she never comes directly to the nest or back directly. We can only find 2 nests for whole trips, and the reason is that we just sitting for the long time same one places, and we see that one female is just flying several times in a one location. It's not exactly one location in a different places, and then we just search it. It's not easy to find.
Because it would be cool to have samples from the chicks to see the composition of the clutching.
Yeah. I think for only one chicks we did the sampling, which is too small. I didn't put the picture. We found the nest, and then we just did the sampling for one of the chicks, and then we put it very quickly in there because we don't want to bother the female, because the mom is always just somehow singing and then flying around of the nest. Yeah, but it's not easy to find the female even. No.
The red-headed one and the black-headed one, they always prefer their own species. So the point that you mentioned that there might be a mitonuclear incompatibility, maybe that might not be the choice because they are preferring different insects, they're preferring different ecological zones. So it's just their preference of their comfort, maybe. So
Yeah.
not even notice the mitonuclear incompatibility.
That's true. That's true. It's the idea behind the second project that we are now working on. We wrote the proposal. Is exactly the idea, because we are thinking that maybe some ecological pressure also play a role in the hybrid zone. For example, different food. If the parents have a different food preference, then maybe the hybrid is in the intermediate and then cannot find the good food, or less chance to find the food which is suitable for the hybrid. I think that's true. Maybe it's some ecological pressure play a role on top of the sexual selection, and then it's the reason that we see such a negative selection against the hybrid. Yeah, that's true.
Okay, so we can stop the discussion for now. And I forgot to say that Niloofar is here thanks to, her visit is supported by this INOLEC program of Faculty of Science. So please don't forget to sign both lists of attendance. It's necessary to do it. And I would like to thank you once again for excellent talk and very nice discussions to all of you. And so thanks once again.
Thank you.